again, we're going to be looking at DNA, going back over that DNA and DNA replication, but then we're going to build on that and look at mutations. So I do want you to go ahead and take out your DNA replication sheet that looks like this. When it loads. So, it looks like this. This is the one that we worked with on yesterday. Now, when we're looking at this, I just want to do a quick recap or a review of what it is that we did.
Well, in the nucleus of a cell.
In the nucleus of a cell. And when we break that down and we get to the nucleus of the cell, then what? One second. We have that DNA, but that DNA is comprised, first, before we see the DNA, we see something else. What is it? Victoria. We see chromosomes. And then we have to look deeper in those chromosomes, and those chromosomes contain our genes. And then those genes break down into these things that look like staircases. And not only just flat ladder staircases, they look like twisted staircases, and we call those double helix. And that consists of what type of macromolecule?
Protein.
No.
Oh, carbohydrates.
No. Nucleotide, good job, you two. And that nucleotide is made up of a phosphate, sugar, and base. Now, if we only said one nucleotide or one side nucleotide, which was the phosphate, where's the phosphate located on that ladder? What would that ladder part be? If it was a ladder, what would the phosphate portion be?
That would be the railing.
The railings, the rungs. What else would make up the railing or the rung? Sugar. And then those steps that you climb on are going to be the what? The bases. And in order to complete your staircase, because you can't have one side of a ladder, that would not be safe. In order to complete the staircase, you have to have the other side. So how do we get that other side? We pair it. And we pair it how? Raise your hand. How do we pair it? Oh, these two are the only ones that know. Nobody ever here knows, Jenny. How do we pair that ladder? With the bases. And what bases pair together, Brennan? A to T. Stop. What's A? Adenine. To T. What's T? Thymine. And then what else pairs? Troy. C and G. What's C? Cytosine. What's G? Guanine. And so those bases pair together. Then we need that other side, which is the what? The other railing we need sugar and phosphate, and we put that together to get our DNA, correct?
Yes.
But then there's some times where DNA has to copy itself. And when DNA is copying itself, we go through two processes. What are they? What are over here? What are they? Victoria? Which one is first? Translation. And then transcription. All right. So in order to do this, what's translation? Tell me in your own words what happens with translation, Anastasia. The DNA is interpreted by using what? mRNA. What does mRNA stand for, Rosalind? Messenger RNA. What is the job of the messenger RNA? Think about that analogy that I gave you. What's the job of the messenger RNA? Simon, when we help you redeem yourself today. No, it doesn't tell you what's wrong. Can't tell you that. It documents what though? Chappie.
I'm sorry, it documents what's wrong.
No, it doesn't. That's what the nurse does in that analogy. But it has to, you're telling it in the doctor's office, you're telling her something, and she has to write it down in a way that the doctor would understand what you're saying. So she has to do what? Interpret or translate. Now let's take that back to the DNA. What is mRNA translating or interpreting? Right. The basis. And why do we have to say that it has to translate or interpret the basis? What no ma'am. That's somebody to tell me. What happens in that translation? Like why does mRNA need to translate?
It means mRNA needs to translate because it doesn't recognize thymine.
Well, if thymine is the base that's already there, it is going to pair it to what? Adenine. But what happens? Where does the translation take place? In the original base, Jenny?
It checks adenine differently, so they use your cell.
Exactly. If the original base pair is adenine, mRNA says, okay, what I translate that to pair with is what? AU. So you think, hey, you! I got to tell you something. That's that messenger. Hey, you! I got to tell you something. Say that.
Hey, you, I gotta tell you something.
Hey, you, I got to tell you something. So that's how you got the messenger RNA. So once it does that, that's that translation. We have that next process, which is what? Transcription. What is transcription? What is that? Somebody over here. We got multiple brains working over here. Let's get your heads working. Come on, baby, baby. What's transcription? Think of that analogy. Think of what the word transcription has in there, huh? That's the major goal of this whole process called what? DNA replication, which is also protein synthesis. So that's the major goal. But before we can get to that goal, before we can get our medicine from the pharmacist, what has to happen in between, Jalil? It has to be recorded. Okay, that's good. Now what? Well, how do we record that when we're talking about transcription? That's good. How do we record? How is it recording it? But how? Tell me that process. What's happening? What's happening? It has everything to do with the what? The basis. So how does that work? Jenny.
It uses RNA polymers.
Okay, RNA polymers is the process that's used, yes. It's going to use that to speed up the process, but then how do we get all the way to protein? How do we get from these bases to protein? Roger. It breaks up into groups. Okay, what are they? No, you're thinking too deep into it. I need the general thing. What if it wasn't CGU? What if it was TAC?
It breaks up into like transmission.
How? Brandon. Right, that does happen. But how do we get to this protein?
Because 3 bases.
Good. 3 bases equals how many? 1 amino acid. 1 second. So it takes 3 bases, there's 1 amino acid. It takes another 3 bases, there's another amino acid. And as we start adding an amino acid to an amino acid to an amino acid to an amino acid, and so on and so forth and so on and so forth, what do we get? Everybody. Protein. Because we take amino acids, what? A bunch of them would be what? A polymer. And that's how we get to the protein. What's the purpose? Why do we want to make this protein? Why is it important for us to make protein? But why? Like function correctly, like what? It influences characteristics. Awesome. I want you to write that down on the bottom part where, on the bottom of your notes. Huh? I know, but that was a very, very powerful statement that she just gave us. At the bottom right here. Protein synthesis is important because, what? Say it again, Fantasia. It influences characteristics. And not just superficial characteristics. Not just, oh girl, you fine. Oh boy, you look good. Not those superficial characteristics, but everything, in which that's what we're going to get in today. But let's just review this chart that we have here, because we did not go over that chart. And before I look at that chart, I just want to kind of scroll up a little bit here. Roger, what's your question?
The analysis part. Did we put translation first or transcription? Because
I made sure we switched the words because you guys are saying transcription and we should have been doing translation first and then transcription. All right, good clarity. When we look at this picture here, that picture is just showing us exactly what's happening. Remember, your DNA splits open like a zipper. And when it splits open, that mRNA comes in and fills those spots by translating, because it's not just going to happen on one-sided base like we did with our liquors. What do you think is happening with the other side? The same thing, because it's going to make a copy of the DNA. So when we get into making copies of the cells, when the cells split, they split from 1 to 2, and both of them must have the DNA. Which brings me to another point that I want to let you know and make sure that we make mindful of. Is the DNA in all your cells the exact same?
yes.
Yes. But why is it that your stomach cells don't do what your brain cells do? Because they all have the same DNA, and they all influence characteristics. Healing. Okay, so what's that called when they're designing performance-specific function? Starts with a D. Differentiation. Very good. They're specialized, so they're differentiated. So what does that mean? Why, again, does her stomach cells not do what their brain cells do? What's happening in that DNA? Chappie. Roddy, you got it? What's happening in the DNA within that cell why your stomach cells don't think like your brain cells do?
Because it was made.
And I mean, if you don't know the answer, that's fine. Just take a guess.
I think because your stomach still has a job to tell you.
So let me ask you. Let me put this in perspective. You come to school. Let's see. What club are you in? Peer mediation. Do you have a job in there? Okay. They're like officers. For instance, let's say in the club you have the president and you have the secretary. For instance, student council. You are the secretary, right? Or the money. You're the money. You handle the money. Who's the president? Okay, so let's say with the student council you have a president and you have a secretary. All right? Let's do president and secretary for lack of sake of argument. Let's do president and secretary. What's the secretary's job? To write down documents. What's the president's job?
To keep everybody in order.
Keep order, right? Can the president take notes?
Yes.
Why doesn't he? That's not his job. So that job has been turned on or off? Has been turned off because he just has that design function to do. Can the secretary give orders?
Yes.
But does she? No. Why?
She knows her
That
place.
job has been turned off and know their place. So with your cells have the ability to do every job because they all have the same blueprint. That makes sense?
Yeah.
But do they do that job? No. Why?
Because DNA is turned on. I mean, it's not turned on.
Exactly. DNA is not turned on all the time for the cells to do that. Backing back up. Translation. Go back to that portion that we wrote because I need you to make sure you have this too in this area here. Translation occurs where? In the cell. Where does translation occur?
In the cytoplasm.
In the cytoplasm. Make sure you have that. Translation occurs in the cytoplasm. The whole purpose of this process for copying DNA is to do what again? Make protein. So where do you think the final process, transcription, takes place? Oh, no! Ribosomes. Remember, protein synthesis happens in the ribosomes. Remember the job of the ribosomes. Very good. All right, so then let's now move down to that chart that I said. That goes next to transcription in that picture that we wrote. And you can even write it in this space here next to that picture. Huh? I'm excited too for you. All right, so let's look here. Base pairing rules for complementary nucleotides. In the first area where it says between the two strands of DNA double helix, G's going to pair with everybody. C, cars in the garage. T pairs with A, apples in the tree. Got it? All right. But we move over here between DNA and RNA during the transcription process. G pairs with C. That's still true. T pairs with, that's still true. But A pairs with U. And what's the thing? A do what? I gotta tell you something. Any questions about that? All right, very good. Then what I want you to do is, I want you to put your next sheet in front of you, which is titled DNA versus RNA and protein synthesis. No, I want you to put that page in front of you.
No, the basis of
We already did that, didn't we? We did that.
no, we did a string of amino acid creates a protein.
Oh, okay, I see what you're saying. Down at the bottom, how many bases do you need? 3 bases of a, what type of strand code? No? What do the amino acids code for? What base strand pair? Is it DNA or RNA? mRNA or RNA? So 3 bases of mRNA strand code complements the, oh, I'm sorry, let me fix that, I apologize, yes. I need you to do this, Chevy, stop just calling my name out. Let me fix that, I apologize. So again, we're here. I love how I stand up on the screen. So, it should say, the DNA strand complements the mRNA strand and codes for amino acids equals. Okay. So it should say 3 bases of a DNA, 3 bases of a DNA strand complements the RNA strand and codes for, not protein yet, amino acids. A strand of amino acids creates a protein. Check it. Yesterday, everybody had to take it. Well, what were you taking notes on the whole entire beginning of class?
No, next.
There were 2 sheets up there. Well, come on. Goodness gracious. Okay, this is the sheet that you want to have in front of you. While we're looking at this sheet, some of you put the wrong information on your DNA needed too. So for instance, down here, where it said, who discovered the shape of DNA? Not Chargaff. Chargaff came up with the rule of the base pairing. That's who did that. Who discovered the shape? Watson and Francis Crick. Now let me give you a little history. Actually, it was Rosalind, a woman. What? What? Why didn't Crick and Watson? They didn't have the power. She figured it out. She worked with them. They got to win the Nobel Peace Prize for it.
Oh, it's in the movies. The space movie with Sir Roger P.
Yes.
So black people basically the reason why it was black women.
Alright, so here, what's going to happen here is we're going to watch a little video. And watching the short video for DNA versus RNA and protein synthesis, what you're going to do is these little boxes, you're going to either label it with DNA, RNA, or both. These are tasks of what happens there. That make sense? So as you're watching the video, you're going to be looking and thinking of these things. You may not catch all 10 of them while we're doing it, but we're going to come back to it and look at it. So I do want you to catch what you can catch. And some of it you all may already know. Okay? All right, so let's go ahead and watch the video. If it were a regular spoon, you probably could have done better, right? Okay, so the fact that you didn't have a regular spoon, some of your spoons were what? They were missing something, right? And that missing of something allowed you to do better or did not allow you to do so well?
Not so well.
So it took away your ability to Function. Okay. So that is what a mutation is, and that's what we're going to talk about. When a mutation occurs, it's a change in your DNA that inhibits or changes the ability of something. And sometimes mutations don't necessarily change the ability, but for the most part they do. But sometimes they don't. They may just change the way the DNA is made up, and so it may change a characteristic, but not always necessarily the ability. So you've just experienced mutations. Who has the regular score? How many did you guys get? 73. So the fact that they had a spoon that was not altered at all allowed them to do a little bit more. Alright? So let's go ahead and put our note sheet in front of us that's titled Mutations.
Making phonies. Jenny.
Now, we do know that what's located in the nucleus? The DNA is located in the nucleus. And when the DNA is in the nucleus, if we move on and we look just a little bit deeper, we do recognize, we haven't gotten there yet, I need you to not yell out. We recognize that we have these little things called, and you get them from your mom and dad. How many do you get from your mom? How many do you get from your dad? Equaling? Not 100. It rattles down to be able to see these DNA, and we talked about that before, so we're going to move on. We understand that the DNA is made up of sugar, phosphates, and bases. Sugar, phosphates being the railings, the bases being the rungs on the inside. Well, you can't get 100% with both of them because they both have to contribute something. The only amount you can do is 23 and 23 something. That's why you have to.
We got like a real large amount of people like.
No, you would have a mutation because there would be something wrong with your DNA. You wouldn't look like them, you would look like something. You look like something. I need you to raise your hand and not yell out.
Is that like twins?
Twins are identical because they have the same exact DNA from your parents coming out of the same
Is that why, I'm not trying to be funny, but I was watching Law and Order and this baby was born from incest and she had like a dysphoria because it was too much of the same DNA?
Yep, that's exactly right. Good connection. Alright, let's move on. No, ma'am. And then we talked about this whole replication process. We just talked about that DNA, in order for it, remember, put that hood off now. Every single cell in your body has the what DNA? The exact same DNA. So that means all of the base pairing must happen exactly the same in order for the cells to be the same. But sometimes things happen differently. But let's review again. If this is the original strand, what does the complementary strand look like?
T, G, A, G, G, A, C, T, T.
Okay, good. So that, no, not for this one, this is just the complementary, because after it does that whole mRNA process, then it's going to decode that back so that the DNA, because remember DNA doesn't have U's. Alright? So then, we move on. The following DNA, the sequence codes for how many amino acids are coded in here?
2, 12, 18.
And how are you getting that?
About 3 bases.
But you just counted 18 bases.
We have 6.
So how many, excuse me.
18 to 5.
That means how many amino acids are located up here?
6.
How is she getting 6? 3 bases equals 1 amino acid. So let's take a look. Yes. Any questions about that review? All right. Then moving on. I need the extra talking to stop. We've already gotten up and done something, so we need to be back focused. All right. So 3 bases code for?
Hold on.
3 bases code for 1 amino acid. Very good. Then moving a little bit further. Oh no. Alright, so in your mutation, I need you to start writing. Mutation is a change in DNA that affects genetic information. Obviously, it's going to affect genetic information because your DNA are your genes. That's your genetics. And we need to get through these notes today. Tomorrow we can do an activity, and then you are taking your test. Woo! I already told you yesterday you had a test, and I sent reminders. And nobody responded. So.
I responded.
You responded with a bunch of colds and.
I don't know why it did that.
Alright. Yes, you can. Alright. When bases are changed, yes. Mutations are when bases are changed, and they're changed by the number of bases. So if the original strand has 12 bases, and the number of bases change when they're copying, and now you have 13, that's going to be a mutation. Mutations occur if the type of bases change. So if you originally have adenine and now it's a thymine there, in the original strand, and in the order. So if it's adenine, guanine, cytosine, and now it's guanine, adenine, cytosine, that order of bases that happen, that's going to cause a mutation. So anything where there's a change in the DNA. All right? Any questions? All right. So type of mutation. For example, you're not writing anything. If we have the fat cat ate the rat, right? But then now we have the fat ate the rat. Something is something is missing. It's a total change in the meaning. Correct?
Right.
All right. So that's an example. Your original sequence that you have here, your paper. You need to look at this. This is what you have. So when we look here, we have A to T, A to T, A to T, C to G, C to G, C to G, all of these complementary strings, but when we go to copy it, meaning they split up apart, and we get to copying, and all of a sudden, we now are missing a base pair of C to G. That's no longer in that copying. Something's been removed. When you take something away, you call it a what? Starts with the letter D. When you take something away, it's not math.
Decrease.
Or, I say start with a D. Deletion, yes. So this is where a base is left out.
That's not math.
Delete. Well, you could. It's still a math term. You may not use it now in the upper grade levels. It's my term. Okay, there we go. Deletion. This is when your base is left out. It's deleted. Or if you're typing and you delete, you're going to remove something. So it's been deleted. Your base pair is left out. So you can put base pair next to a base left out. All right, here's another example. The fat cat ate the rat. Fat cat XLW ate the rat. So now we have something different. Something was added. So in the example, your original base pair here, and then we look at our new base pair, and now we have an A to T base pair in our DNA. So here we have a base pair was added. What are you saying? So I want you to put, where it says an extra base, put extra base pair added. Because what's going to happen, you're going to get another base, which means that the copying is going to require another pairing of it. And this is called insertion. Think of your computer terms, I guess. That makes better sense. So you have delete to get rid of, insert to add. So notice so far these are all changes, right, in the DNA. Remember, it codes for an amino acid, which codes for a protein. So if we look at this before, the AAA would have been coded for a UUU for a specific base. I mean, a specific amino acid, correct? And then you would have had the CCC would have coded with a GGG and mRNA for another amino acid. But in the copying process, you have the UUU, but now you have a GUG, which is not going to be the same amino acid for GGG. Remember, all of your amino acids are going to link together to create a protein. So if those amino acid pairing did not come together correctly, instead of having maybe Instead of maybe having a salivary protein, a protein that's going to create salivary glands to help produce saliva, it may code incorrectly where maybe you now can't produce saliva. That's a characteristic. It could be possible. That's a characteristic. A chain. That's the protein are for characteristics. So see how that could be a problem.
Could you promise not? You know how people like have artificial
Yes, I
tears?
did. Sometimes that happens. All right. So here we have the fat cat ate the rat once again. But now we have the phrase the fat hat ate the rat. Total difference. Definitely don't make sense. So here, what has happened?
Change the way.
It was changed, not just the word, it's like a change of the letter, not the order. Something wrong or something, not necessarily wrong, but something different. So like for instance on Tuesday, I wasn't here, there was someone different, you had a what? Substitute. And that's what this is. Here you have the same amount of base pairs, but now you have something different. Before you had less, the second time you had more. Here you have the same amount, but there's something different. There's a change in the base pairing. So you have the wrong base pair used. And that's going to be substitution. Yay! Any questions on that? All right, so now you're going to try. On your own, on your notes, you have this. If DNA strand 1 sequence is this, what should DNA strand 2 be? Go ahead and do that now. That's the complementary strand. All right? Let's check your understanding. You should have selected this because we know that apples in the tree, cars in the garage, and that should have been all well and dandy. Now, the process of the cell cycle is when your DNA actually copies itself in the process of interphase, which we'll talk about after midterms. So strand 1 and strand 2 is translated to having this. Because after the translation process, it's going to come back together and create the actual DNA that needs to be within the nucleus. So if we had this during the translation as opposed to what's on the left side, what type of mutation has occurred? Substitution? Well, let's see. We have 1 base pair, 2, 3, 4, 5, 6, 7, 8, 9, 10 on the left side. On the right side, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So we know that it cannot be insertion. It definitely can't be insertion. It definitely can't be, definitely can't be deletion, and it definitely can't be substitution, because substitution is the same amount. Deletion means a separate amount. Insertion is going to be more. So, and there is more. So here, it definitely is insertion. All right? So now, and this would be considered a point mutation. A point mutation. Alright, so there are 2 ways in which DNA can become mutated. One way is parent to child. Mutations can be inherited. So inherited means that it comes from a parent. This is just something that was already in their DNA, so you got that same DNA. Another way is mutations can be acquired. What does acquired mean? Obtained. It can be picked up somewhere. And not like you can go to the store and buy it. But acquired means there's just a mistake when your DNA is copied. So your DNA copies in a mistake. Now this happens all the time. But our DNA also has a corrective process where it actually corrects itself. Thank God for our DNA being able to do that without us controlling it or even knowing that it happens. DNA can be acquired through environmental damage from mutagens, and we'll talk about what mutagens are in just a second. So mutagen is anything that causes a change in DNA. Everyone good?
Yeah.
All right.
Example?
So, some examples. Asbestos. You've seen the commercials if your family has suffered from, what is it called? Mesothelioma, please call because it's from asbestos. Now, what asbestos is, it is a substance that is in the walls, in the insulation of old buildings. They used it. Now, when contractors have tore down old buildings, they become exposed to this. It only is harming once it becomes airborne. And that exposure is a mutagen because it's going to cause a change in the DNA. Mutagens also cause cancer. All right, cigarette smoke. All right. X-rays are mutagens. This is why they tell pregnant people that why they limit the amount of X-rays given to pregnant people because it could create a change in the DNA for the baby. Because they're developing a baby, that baby is copying some of the parent's DNA and it could change it. And this is why they don't want you, every time something's wrong, they don't give you an X-ray because it's a mutagen. It's X-rays. UV lights.
Oh, there's what people who do like chemo, they wear things.
Yes, and also getting tanned. When you do get your nails done, you do sit them under UV lights. And definitely doing that. All right. We're going to use the following terms, and right now I want you to take about 20 seconds and use the terms mutation and mutagen in a sentence. Do it now. Ma'am. Why you? Why you two?
I can't write.
Let's do it now. Create a sentence using these two terms. We're doing really good. But I do want you to see sentences.
She said environmental damages from mutagens.
Environmental damages, yes, from mutagens. And those mutagens are all the things that we just, well, not all of them, but the things that we just listed. All right, give me a sentence, Kanar, since you're talking. You must have a sentence for me. Don't put yourself on the spot. Chappie, give me a sentence.
Mutation is a change in DNA that could be affected by mutagens.
Not affected by mutagens.
That
can be obtained from mutagens. So mutations are caused by mutagens. Simple sentence. I'm moving on. Mutagens are anything that causes a mutation. That's another sentence. All right. Significance of mutations. We want to understand why this is important. Why mutations, knowing what mutations are, how they happen, why that's important. Some of the things, it could be corrected by DNA repair, meaning that it could happen and DNA is automatically going to kick in and say, I got it, don't worry about it, you'll never know about it, I fixed it. Something's not right, I fixed it. Mutations can be beneficial. They're not all harmful. One type of beneficial mutation is some people develop a change in their DNA which causes them to be immune to HIV.
Oh.
That's a benefit. It doesn't happen with all society, but that's a benefit. Hold your question. 1 second, hold your question. Mutations can be deleterious. What word do you see in deleterious? Delete. Normally, delete means to take away. Well, it does mean to take away. When something gets taken away from you that you want, is that a good thing or a bad thing?
Yeah.
That's a bad thing. So what does deleterious mean?
Taken away.
It means bad. It's harmful.
Oh, bad.
So some mutations can be deleterious or harmful. For example, sickle cell anemia. that is a change in the DNA, because all of your blood cells should form like a normal healthy blood cell. But if they turn into a sickler shape, that means that now the shape has changed. They can't, they don't have enough surface area to produce that oxygen. To carry that oxygen throughout your body. So you have low oxygen levels because your cells not the correct proper shape. Also, if you look at this picture here, they start to get clumped up when they are flowing. So not only is oxygen not flowing to your body, it also can form blood clots because they're also clumping up in one area because they have to fit through those capillaries.
Okay, so I asked Mr. Lee because I told him that both my parents got sickle cell, and he said that my blood cells look like that, but I told him I don't have sickle cell, I have a trait.
You have the trait, that yeah, you probably just have the trait.
I do.
Yeah, you don't have it, you don't have
so
it.
So my blood cells don't look like that?
No. You have a trait, which means that you would pass that trait on, you could pass that trait on to your child. Now, if the significant other that you marry, I'm going to talk about this later, we're going to get more into this, that you marry has a trait, it's a possibility that your child could have it. Or if this person that you married has sickle cell, then it's going to be a possibility that your child can get it as well. No. It's passed on with it's a trait. This is one that you've inherited from your parents. All right. Mutations can, one of the deleterious mutations is Down syndrome. That also is a extra chromosome. That's going to be an extra chromosome in your DNA, and individuals who have Down syndrome usually have learning disabilities. So we have ones that can be beneficial, like giving immunity to individuals, deleterious, like causing non-passable diseases or non-communicable diseases, and another one, they could be harmless, meaning you can have a mutation and it's very harmless, eye color. Some people's eye color, not like having blue eyes or green eyes, but sometimes you have someone that has a brown eye and a blue eye.
I've seen a little girl.
Yeah, the little cute little twins on Instagram. But she sees perfectly fine. It doesn't change her eyesight. Because remember, every part of your body is developed from a cell, and they all copy the same way, which means that your eyes should look the same. And when they don't, that's a mutation. But this doesn't harm you. Another type of mutation is birthmark. How many of you have one? Mutant, 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 mutant. Because again, all of your skin cells should look exactly the same. And when they don't, it's a mutation. It's a change in the DNA.
So if you don't work, that means there wasn't a mutation that happened.
In your skin cells, right?
That means you don't have a mutation.
Yep. Extra finger. Some people do grow, this one is a little bit more profound, but sometimes people have a little pinky, or it's just a small piece of skin, because again, your skin cells are supposed to copy exactly the same. Guys, pay attention. Your skin cells are supposed to copy exactly the same, which means you should have 5 fingers here, and you should have, 4 fingers on each hand and a thumb on each hand, but when you don't, then there's a change. Let's consider the thumb. How about this? Because we need to move on. Yes, well, that's going to be, yes. We'll talk about that a little bit later also. Albinism is also a harmless mutation because it's a change in the pigmentation of the skin. It's harmful though, harmless. Although some people that have albinism, they cannot be in the sun as long because they don't have the melanin in their skin to protect them from the rays of the sun. All right. So, for example,
It's like vitiligo.
that's not like vitiligo.
No, not about it, I'm saying. I want to say true.
No. Hold on. This is how we get my attention to ask the question. Give me 1 second because I need to. That's not what I want to do. I want to.
Ask your doctor.
All right. So on your notes, you want to make sure you study mutations and how they occur and all of that great stuff with it. But just looking at some examples of albinism, just so that you can see that it's not just individual organisms that happen. You could have, first of all, what we're not going to do, number one, what we are going to do, I should start off with positive, is Troy. we are going to be mature, because what you're exhibiting right now is prejudice. Because something looks different to you, you're going to make fun of it. And isn't that what you don't want people to do to you? Hello? So just because you may not have ever seen anything like this and it looks different, does not mean, number one, you're going to make fun. Number two, also does not mean you're going to start picking people out and trying to identify them with what you see that looks different. Because that's very immature and it's rude. So not just humans can have albinism, but animals as well. And we'll talk about how it occurs a little bit later when we get into genetics. Yeah, when we get into all of that, we'll talk about that later. While this is restarting, what I want you to do is put your lyrics in front of you, because there is a song that will help you with albinism. The first thing that Alright, so there is an albino snake, there's an albino turtle, you have an albino alligator, there's also albino lobsters, all organisms. Then you have the cat, there's a pug named Vicky, a hedgehog, and there's an albino young lady named Bianca. There's an albino butterfly, a cockroach, there's grasshoppers, and then even flowers. And notice that it's not just always white, it's yellow or reddish pigment because it's missing the actual pigment, colorful pigment that it has. Alright, so go ahead and put your lyrics in front of you. No ma'am, no ma'am. No ma'am. Alright, the first thing we're going to do on today is we are going to fill in the blanks. Roger, hold your question for a second. We want to get this in, and this is what you're going to use to study. So it goes, blame it on the gene if your eyes are green. Quiet. No, ma'am. Notice it talks about, we got to get through this. Notice it talks about genes. That's located where? In your DNA. So that's why it's talking about blaming on the genes, because your eyes being green is all about your genes. 6 fingers on your hand, blame it on the strands. I want you to underline strands. What type of strands are we talking about? The strands of DNA. You might want to write them on the side. If we have extra talking, we need to stop. So on the side, you want to write the strands are referring to the DNA. Blame it on the DNA, blame it on the adenine. Why are we mentioning adenine? It's all, and copying of the DNA has everything to do with the bases. And you know it's in the helix. What helix? The DNA helix. Hydrogen bond helix. Why are we talking about that? The bases are bound with strong hydrogen bonds. You might want to make sure you know that.
I'm going to listen to it.
Are you going to write down your lyrics? But when it gets unraveled, it makes mRNA. What's that unraveling?
The splitting.
Which is what process? Transcription. mRNA travels from the nucleus to the cytoplasm. Remember, that happens in the cytoplasm. So you might want to put to the side, transcription happens in the nucleus, in the cytoplasm. Ribosomes, it has them translate into amino acids making protein. That's that whole translation. Nitrogen base dictate your races. That's what nitrogen is base. As A to T, C to G. Transcription, translation. You make mRNA from your genes all day, then translate it into polypeptide chains. Again, you learn the song, you know the work. Of which you got a lot of. The central dogma is basically this whole study of understanding this copying of DNA. DNA to tissue can be quite an issue if you have a genetic disease. Because a genetic disease means that's going to change your DNA.
Who made it?
First of all, let's just do this. We don't have time to stop and ask this unnecessary questions. Blame it on the rungs. What are the rungs? That's the ladder, the middle pieces. You might want to put that on the side so you know what the rungs are. That's the middle piece. Even if you can't do this, that's because of DNA. Blame it on the base pairs if you got some red hair. The pairing of the bases. Blame it on the DNA. Blame it on the thymine, another base. Blame it on the chromosomes. Blame it on the helix. Again, DNA helix. If you're hemophilic, I'm sorry, I missed something. Blame it on the DNA, blame it on the guanine. I did, it's right there. If you're hemophilic and you can't heal it, we'll talk about hemophilic later. UV rays can mutate, that's just in your notes. Replace a C for a T. That's a point mutation. The change is final. You might be an albino. Depending on what gene gets messed, what protein is expressed. Again, like Fantasia told us, proteins are our characteristics. Nitrogenous bases dictate your races by protein we make in transcription, translation. Sorry. And then again, the same thing. You make mRNA from their genes all day, then translate into polypeptide chains, of which you got a lot of. That's the central dogma. DNA can be quite an issue if you have a genetic disease. See y'all later. Bye-bye. Study, study, study. Tomorrow we'll do some review and then we'll take a test. Huh? Tomorrow we'll do some review and then you'll take your test.
So, it's better life is better life.
It's a disease that happens. And we'll talk about that later when we get more into genetics. I'll get it. No. You know what? You don't put No, it probably changed. Like some people's eyes when they were born, they were fully developed eyes. My son was gray eyes. I